So welcome everybody. Today's UCSD Prepare Institute seminar is going to be by Dr. Kline. Next slide, please, David. And she's going to be talking about next generation sequencing for pathogen detection in bronchial lavage specimens. She's an assistant professor in clinical paths. She's also an ID doc, an excellent one, and associate director for our micro lab for our students and for our hospital. So very pleased to have her talk today. I think there's another slide for me. And then just about her, she's an internal medicine resident here at UCSD. I remember those days well, and then became the first chief resident in quality and safety at the VA. And then she left San Diego and completed two fellowships at the NIH, both in infectious diseases and in clinical micro. And she continued working at the NIH as a staff clinician until she received, until we recruited her here. To run our labs and teach us all sorts of great stuff and do research and take care of our patients. And a couple years ago, she became the associate director of our clinical micro lab. So it's very awesome. And it is my privilege to have her talk today about BALs. Take it away, Dr. Kline.
Okay, let me just share my screen. Okay. People should be able to see the screen. I'm gonna just make you all small. Can you see my screen and see my laser pointer, hopefully?
See the screen. Yep, the laser pointer too.
Yeah. As you saw, this is NGS, or next generation sequencing for pathogen identification in BAL samples. I think somebody must be like slamming doors in the hallway behind me, so I apologize in advance for any noise. Okay, so goals for today, or the goal in general for this project, and it is still ongoing, is to develop a methodology for untargeted next generation sequencing, NGS, from bronchial lavage specimens. And the idea is to elucidate novel pathogens and just get better elucidation of existing pathogens, and to develop it in such a way that it could also be developed into a clinical diagnostic test. So I'm going to talk a little bit first about sequencing evolution as it relates to two pandemics we can all relate to. Well, maybe some of us more than others, for the 2003 SARS pandemic, and then of course the current COVID-19. Discuss the existing sort of NGS diagnostics that are on the market. We're not the first person to go in this direction. We're actually, one of many labs thinking about next generation sequencing as a clinical tool and as a pathogen detection tool. And then the progress on this project, and then next steps and future direction. So just to give a little bit of background, the first severe acute respiratory syndrome, or SARS-1 as we might call it now, started in 2002 in November, when the first case occurred in Guangdong, sorry if I'm mispronouncing that, China. It wasn't until February 11th of 2003 that the WHO was notified of 305 cases. And so a few months had passed where the virus had been spreading and kind of the world didn't really know. By March 11th, so a month later, there had been reported spread to Hong Kong of the first SARS-CoV-1 virus, as we call it now. And so now, obviously, there was more global concern because the spread had sort of moved beyond sort of one region of China. In April, just a few weeks later, on April 2nd, 2003, the WHO issued a travel advisory sort of for people traveling to areas where they knew the original SARS syndrome was occurring, and then also for countries to be aware of returning travelers from those areas. So there was airport screening and a number of things in the US. And then it was identified as a coronavirus at this point, right? It was just thought to be a virus that caused an acute respiratory syndrome by microarray on April 9th, 2003. They actually had some data a little bit before that likely said it was a coronavirus, but we'll use the April 9th day. So that was now about 5 months later after the original first cases, and certainly 2 months after the WHO kind of found out about the virus. And that was thought to be pretty groundbreaking at the time, that we were able to identify it so quickly. And by we, I'll tell you who, not me. In June 2003, it was declared under control. So fortunately, this pandemic did not last a very long time. And because of that, probably because of how sort of quickly it fortunately burned out, there was no readily available clinic diagnostic test ever on the market. And that is because probably the need to kind of have a readily available PCR for this virus kind of disappeared. So I'm sure one could have been developed if it had continued. As I said, the original SARS-CoV-1, as we call it now, was identified by microarray. It was sort of published as a coronavirus officially on May 15th, 2003, but of course there was pre-publication before that, as I mentioned, in April. And it was identified by Joe Dorishi and Charles Chu at UCSF using a virochip, which was a microarray technology, comparing fluorescent signals, and it sort of lit up the coronavirus region or blitz on that microarray. So let's contrast this with COVID-19. So we all know this story very well. So I'm just going to point to the parts about diagnostics. So we all now know that the first cases were described in late November, December 2019 in the Hubei province of China. On December 31st of 2019, the WHO was notified that there were cases. At that time, of course, they thought they were associated only with this Hunan seafood. On January 10th of 2020, so about 11 days after the WHO was notified, the GenBank upload of the viral sequence was done. So that is a contrast in the identification of the original SARS-CoV-1 as a coronavirus, which was many months after the first case and 2 months after the WHO was made aware. This was within days. And I think that's something that tells you what has changed in 2020 compared to 2003. On February 4th of 2020, the FDA issued its first approval for the CDC diagnostic kit. Of course, only the CDC had it. On February 29th, so a few weeks later, the FDA, not the CDC, allowed labs to test prior to EUA approval. So you had to apply to the FDA for your test, but you could go live with your test before you got that approval because they knew there was testing need. Well, I was not here at the time, as you all heard. I didn't get here till fall of 2022. But on March 8th, 2020, the first UC San Diego diagnostic PCR was available for our patients. And I think that's a big change, right, compared to what we saw with COVID-1. So why did this happen? Well, next generation sequencing. So it hit the market really in the mid-2000s and also resulted in reduced sequencing costs. So most people are aware of Sanger sequencing from the 1970s, and then the amount of dedication of money and dollars that it took to describe the whole human genome in 2003. And then synthesis-based sequencing occurred in the mid-2000s, Illumina sequencing, ligation, which was called solid sequencing, eventually PacBio, and then Nanopore by the mid-2010s. And so there was kind of a blossoming of sequencing technology that occurred really in the mid-2000s. And if you look, this is a figure from the National Human Genome Research Institute at the cost per raw megabase to sequence DNA. Even if you follow Moore's law, it's beaten Moore's law. And you can see that that real exponential drop-off starts to occur in the mid-2000s, which seems to correlate with sort of the ability to do sequencing more robustly and more cheaply. And so now we're down, over here where the cost is one billionth, I think, of the original cost to do the genome. Okay, so many people on this, I think in the audience probably are aware of types of next generation sequencing that are commonly used, obviously. And there are three here kind of worth highlighting because we had to select what type of sequencing we wanted to try to do this assay. And so I would say the big contenders were Illumina sequencing, PacBio, and then the Oxford Nanopore. And they have different pros and cons to them. Some can do longer reads than others. So you can see that the PacBio and the Oxford Nanopore are able to do longer reads than Illumina sequencing. Illumina is able to do a lot of reads. The processing time varies, the error rate varies, with Illumina kind of winning there. The cost per run varies and the instrument price varies. And so, your advantages are your Illumina is a highly accurate sequencer, and your PacBio and Oxford Nanopore sequencers can do long reads, and then the Oxford Nanopore has the additional advantage of being a portable device. So the first thing that mattered to us was the cost per run and the cost per the instrument. So getting, I am a new assistant professor who's a health sciences faculty who spends most of my time on clinical work. And so, I don't have a grant to buy $1 million instrument. Additionally, where there are already many great people on campus who have an Illumina sequencer, for example. So the instrument price and then the cost per run price of the nanopore was attractive. And then the other thing, of course, is the processing time that is short. So this was really designed to be kind of I would say the most user-friendly sequencer, even at at a little more error rate potentially. So nanopore sequencing, for anyone in the audience who doesn't know, it is kind of a brilliant sequencing technology where there's a pore, a constructed nanopore in a membrane, and flow of single-stranded DNA moves through that pore, and across that membrane, there's a voltage being applied. And as the piece of DNA sort of clicks through the pore with different nucleotides, each base changes the current across the membrane, which can be measured by a sensor chip. And so based on sort of the base calls are made based on current. And this technology has evolved both in the read accuracy and the speed of read over time, really from its original conception in the 1980s to sort of its current fungal and I think I think on this sequencing did the R9s, but we're at R10.3. The analysis can even be performed on a laptop, although you should certainly get a pretty high processor, which I'll talk about, a good processor as which is important for excluding human reads. So why does UC San Diego need a sequencing-based diagnostic? So there are a couple of things, and these are real things that are happening right now. So one would be difficult to test or culture pathogen. So we recently, this is an actual screenshot from an actual patient had a patient with endocarditis at UC San Diego. And one of the organisms that can cause endocarditis is Bartonella. So Bartonella henselae, which causes cat scratch fever, and Bartonella quintana, which causes trench fever, both have been implicated. And the current growing Bartonella is very difficult. It's almost impossible for a regular clinical lab to grow it today. And so most people end up making the diagnostic and diagnosis in one of two ways. They get serology because the patient will usually have had Bartonella for quite a while. So they'll have an antibody positive, but that antibody may not be specific to a species of Bartonella. It can be hard to tell. Or they may get a PCR, which is available at our reference lab, not here on campus. That PCR is also not species specific. So this is a sequencing-based technology that I'll talk about later that actually identified it to the species level as cat scratch, Bartonella henselae. Also, we have for monkeypox virus. But before this, the kind of sort of epidemic monkeypox that we have been dealing with in the last 2 years has been clade 2 of monkeypox, which is thought to be less virulent. And now there has been clade 1b, the more virulent monkeypox. So potentially an emerging pathogen detectable diagnostic might be able to distinguish the clades from each other if you could get enough of the reads of the sequences. All right, so what exists on the market that's currently used by our healthcare providers at UC San Diego? So one of the most common sequencing, next generation sequencing based diagnostic that is ordered by providers here is the Carius test. And it uses sequencing in blood, and it looks at cell-free DNA in the blood. So they take the cell component, it's spun away, although you have to do that rather quickly, and then the cell-free DNA component is put on an Illumina sequencer. And so it's DNA, and so in theory, it can detect any pathogen DNA, including DNA viruses. That have DNA in the blood. So obviously if the virus or the bacteria or the pathogen is in the blood, you would expect their DNA there. But even if there's leaching of that nucleic acid into the blood, one can detect it there. It misses RNA viruses because they don't do RNA sequencing, and infections where there isn't DNA from the pathogen leaking into the blood. So that, more localized or walled off infections that haven't had blood penetration of the pathogen, or at least nucleic acid from the pathogen, won't be detected. They report a turnaround time, that's what TAD is, of 2 days from specimen receipt. Our actual observed turnaround time here is often 4 to 6 days. That's because we have to get them the specimen and then get the result back and put it in the record. And importantly, it's a $2,000 test. Actually, they just raised the price to $2,200. It's going to go up in October, I think, for anyone who orders the Carius. The next probably most commonly ordered sequencing, or maybe third most common actually, diagnostic ordered by UCSD providers is the UCSF metagenomic and next generation sequencing. And so they used to only accept cerebral spinal fluid, but they'll also accept plasma. And they do unbiased sequencing of all DNA and RNA using Illumina sequencing. And so in theory, any DNA or RNA pathogen in either the cerebral spinal fluid space or in theory the blood, so they may have a benefit over the Carious test. So what can it miss? Well, if an infection is not in the cerebral spinal fluid, then you won't catch that infection by sequencing CSF. And then similarly, infections without nucleic acid spillage into the blood. So if it's not a bloodstream infection or significant infection where the DNA or RNA is making it into the blood, then they'll miss that. They report a 1 to 2 week turnaround time for their assay, and so much slower than the Carious assay. It is often about 2 weeks from the time we spend the specimen, sometimes a little bit longer, and they charge $3,900 for their test. And then the University of Washington offers sequencing as well, based at diagnostic. They'll receive any sample type, which makes them sound very exciting. But instead of doing unbiased, sort of sequence all the DNA or RNA that you can find, they do targeted pathogen type detection and use an Illumina sequencer for it. And so it looks for DNA from either bacteria, mycobacteria, or fungal pathogens. For bacteria, their target is the 16S, our ribosomal RNA region. For mycobacteria, it's the SEC-A and RPOB regions, as well as the 16S region. And for fungal pathogens, it's the ITS sequence. They miss RNA viruses in their pathogen, in their samples, and they report a 5 to 7 day turnaround time from receipt by University of Washington. But in our actual practice, it ends up being closer to 2 weeks, again, accounting for receipt to our lab and then sending it out to them and getting the result back. It's $1,200 if if you're hoping to get all 3 possible pathogens detected. At present. So it's the cheapest test, but also the most targeted. So if we want to build a sequencing-based diagnostic at UC San Diego, we want the ability to detect pathogens in an unbiased way, because that also allows the opportunity to potentially find an emerging pathogen. We want our turnaround time to be better than our competitors, and we want the cost of testing to at least be equal or ideally better than those other sort of existing sequencing-based diagnostics. So our objective is to develop a next generation based assay for detection of pathogens in bronchial lavage specimens. We chose bronchial lavage specimens because of the recent COVID pandemic and the idea that respiratory viruses seem to be the most likely new and emerging pandemics that we're going to see. And so looking at the places that they infect seems the most useful. So step 1 is preliminary assay development and workflow, and then early sensitivity and a little bit of technical reproducibility data, which is what we'll talk about today. We've optimized the assay with DNA pathogens, mostly bacteria, and we've sort of started with RNA viral pathogens. Eventually then to complete a full analytic validation, which is necessary to bring an assay into a lab, and then follow that ideally with a clinical validation in the future. But in the beginning, you only get enough money to buy the flow cells for the first part, right, to optimize the assay. And so this was our plan. The idea was to use 10 flow cells for RNA viruses, 10 for DNA viruses, 10 for bacteria, 10 for specimens with bacteria and viruses, and then 5 negatives. So what DNA pathogens could we look at? So we have clinical samples provided by the clinical lab that have positive growth of bacteria, which obviously will have bacterial DNA. And then we also have clinical positive PCR diagnostic tests. And so specimens that were positive by that PCR test, which I'll describe in a few minutes. And then additionally, BAI Resources, which is actually an NIH repository, has DNA from pathogens that could be spiked in. So we reached out and ordered from them DNA from Listeria, Vaccinia, Cowpox, and Yersinia pestis. The idea being that, if you want to show that you can detect an emerging pathogen, you might want to spike in the DNA from an emerging pathogen. And as fun as it would be for me able to work with Yersinia pestis in the lab, we're not allowed to work with it because, we are not at the level of biosecurity here for us to work with the actual pathogen. For RNA pathogens, again, clinical samples positive for RNA viruses would be the more, unique. Obviously, your DNA pathogens are transcribed into RNA, but there are viruses, obviously, that live in the RNA world. And then RNA from pathogens from BEI. So these are the ones we tried to order. We have Corona, Ebola, and then Lassa virus. And then we also were lucky enough to get from a few PIs here, Dr. Carlin, Dr. Steinmetz, and Dr. Lewinsky, some either heat inactivated or not inactivated in the case of tobacco mosaic virus, or detergent inactivated viruses of standard sort of respiratory pathogens, tobacco mosaic virus, and HIV. Just to put out there the current UC San Diego diagnostic tests for bronchiolar lavages that exist. So we have standard culture methods. This is just an example plate. You can see that often there are multiple different bacteria that grow on the colony cultures, and they have to be individually identified and subcultured and subsusceptibility tested. This is also a regular sheep blood culture, but if you want to grow, say, a mycobacteria or a fungus, then you have a much longer turnaround time, as those take longer to grow and have to be put on special media. At UC San Diego, we no longer do viral culture, thanks to the development of PCR-based diagnostics, which are more rapid than trying to wait, say, a couple of weeks for a specific herpes virus to grow. So we do not have that as a standard culture method. The other thing for a bronchial or lavage specimen that we have is the respiratory pneumonia PCR panel that's made by bioMaru, the BioFire panel. It has a number of bacterial targets, including ones you would expect, and then a few that could be cultured. So pretty much everything from strep pyogenes, with this group A strep, all the way up here can be cultured. But chlamydia pneumonia, Legionella pneumophila, and mycoplasma pneumonia are difficult to culture and not typically put into media that a clinical diagnostic lab would normally culture them. And pneumonia takes over a month to grow. There are also adenoviruses in there. Adenovirus is the DNA virus, and then coronavirus, it's metapneumovirus, rhino, and taro. The primer set cannot distinguish them. Flu A, B, paraflu, and RSV. So this is, of course, a rapid diagnostic, but it's limited to the targets that are there. So we are not the first lab to start working in the BAL space. So the University of Washington offers any specimen type as one of their, targets, and they will accept BALs. And so they published a paper looking at, again, targeted 16S rRNA from BAL. This is just an adapted table from their paper, but you can see that predominant by culture, there were a lot of things that grow, and then predominant by that direct 16S sequencing, they found a lot of some other things. There's way more specimens in their publication. This is just a sample of them, but it's to highlight a couple of things. They found a number of anaerobes in their specimens that wouldn't have been easily cultured in standard respiratory culture media, or maybe not identified as pathogenic by the lab. And then they found some unusual but clearly pathogenic organisms like T. whipplei, which is very difficult to culture and also very difficult to stain and diagnose. So their conclusion was that perhaps we are missing anaerobes either in our current culture methodology that are contributing to disease, which is well described, especially in cystic fibrosis, and or sort of pathogens that we would never detect in the first place, like T. whipplei. So this is data from our work. So this is some example data of the first sort of runs of UC San Diego next generation DNA sequencing. So here's what the culture from that specimen resulted, the pneumonia PCR resulted, and then the nanopore sequencing results, classified, sorted by bases per million sequenced, and then with coverage breadth also there. So here you can see, for example, a patient who had a large amount of Moraxella growing in the culture, as well as detection by PCR, and then a Staph aureus with less growth. They also had Aspergillus galactomannan positive, but I don't believe grew it in their fungal culture. Those are just here in the data for points of discussion. And then their PCR panels also detected those same pathogens that grew in culture. So yay, the PCR, I suppose it's a good diagnostic test, as well as some resistant markers. And then in nanopore sequencing, a number of organisms were described that weren't identified in the culture or the PCR, either because they're not on the panel or because they wouldn't have been selected by that technologist, even if they grew. So one was Lactobacillus gasseri, which is potentially a probiotic maybe the patient was taking, which had the most coverage breadth. Moraxella was identified, Serratia was identified, Staph aureus was identified, but with very low coverage breadth. And then Gamella was identified, which is a known oral bacteria. Again, with Pseudomonas, we saw similar things. Here you'll see that rhinovirus and enterovirus was positive, but this is an RNA virus, and this data here that I'm showing is only DNA sequencing. And so again, Pseudomonas was able to be detected along with a number of other bacteria at much lower coverage breadths. Klebsiella was detected in the specimen and then detected by our sequencing, a Chromobacter, and then detected by sequencing. So I bring these examples up just to show that we did seem to, in the earlier runs, be able to detect the things that came out in our known cultures, but also a number of other organisms that might not have been as easily cultured, like Prevotella, which is an anaerobe, and Mycoplasma, which is difficult to culture. One of the problems we had ran into early on it was that huge amounts of human reads were in our sequencing. So the first sample we did, which is here, is called BAL1, had 2 million reads and 97% of it was human genome. Now, nanopore sequencing software can remove the human genome while you're sequencing. And so when you do that, you do enrich in the non-human reads that you're sequencing, but still in the 90% of human reads. Not shown here, we tried saponin treatment and DNase treatment before, different types of purification protocols. Nothing really improved it more than just doing read removal. So except for buying a faster computer, which helps the sequencing happen faster and the read removal happen with a little bit more speed. So then it became time to say, okay, well, for DNA sequencing, can we spike in the DNA that we have from things that we can't necessarily grow in the lab? And so the first thing to do is look at the concentration of the DNA that we obtained from BEI. And many of them were too low to read on a qubit, basically, except for the Yersinia pestis, which of course is a bacteria, whereas these are a virus. So and so there was more genomic DNA available of the Yersinia pestis. So we then took one of our already known sequenced specimens, the Achromobacter specimen, which here's the sequencing, and then spiked in Yersinia pestis DNA and as much of the Vaccinia day as we could. The Yersinia pestis was able to be detected by sequencing with a decent basis per million coverage detection rate, but the Vaccinia DNA was not. So that was kind of our first that thing that we don't expect we would find plague in a standard BAL at UCSD. Sort of, it wasn't there hiding, but known to be spiked in. For RNA viruses, as a start, we spiked in the donated RNA viruses because the concern was that if the viral genomic blocks was so low for DNA, probably the RNA ones were low too. So this is when we reached out to other labs on campus and said, hey, do you have any viruses that you could give us? And as I said, a number of labs did. So thank you again, Dr. Steinmetz, Carlin, and Lewinsky. And the viruses we spiked in were SARS, RSV A and B, flu A and B, HIV, and tobacco mosaic virus. We were able to detect all 5 viruses, although the most detection of tobacco and mosaic virus, so much so that it really overwhelmed the other viruses. We additionally, I will show you, were able to detect the rhinovirus in this specimen. That was the specimen it was spiked into. Tobacco mosaic virus in general dominated many of our future samples because of the sheer amount of DNA of its genome, despite trying to reduce its concentration and set us back a little bit as it just overtook a lot of the sequencing. So the first question you ask is the accuracy analysis. Did we find what we know is already there? So these are the sort of specimens we've been able to analyze for accuracy thus far. And so most of which have been sequenced for only DNA, and then some that have had DNA and RNA, and then one with RNA only. In general, we have detected the things that are there, although we decided to set a coverage breadth of greater than 1% as a cutoff. And so that means the Staphylococcus aureus that was detected technically didn't get detected with that cutoff. There were some examples like the adenovirus, which is a DNA virus that wasn't detected, and the vaccinia, which I mentioned probably was spiked in at too low in the gene block. And there were some examples of bacterial pathogens where only the DNA of the pathogen was detected and not the RNA. But promisingly, when we spiked in all of the respiratory viruses that we were donated, we were able to detect them all. Preliminary analysis of all of the runs we've done so far suggests a DNA target sensitivity of 76% and an RNA target sensitivity of 81%. We haven't focused on precision yet, but so far we've had a few where we've repeated them, and so far they've repeated with 100% sensitivity. So how does this early sensitivity data compare to the other commercially available assays out there? So the Carious cell-free DNA blood test and their validation, analytical validation, they had some specimens that were positive by initial culture and some positive by, quote, all microbiologic testing. And they had 93% agreement with culture and 84% by all microbiologic testing. So potentially better than what we were seeing. But again, they're looking at just DNA and in just blood where things are going to be more pure. UCSF, which does a more unbiased sequencing similar to what we are hoping to do, they, for their cerebral spinal fluid, their initial lab validation data showed they actually separated it out by pathogen. And I was surprised to find out that their sensitivity for RNA viruses was only 67%, DNA viruses was 85%, and bacteria was only 64%. So the sensitivity being on the lower side in the 70s or 80s as a preliminary result may not be actually as bad as one thinks it is. Fungus and parasite, we haven't really attempted yet to look for, so hard to know how we'll compare. So their overall was 73. We don't have data yet to do specificity analysis, so really only able to compare the sensitivities. So one of the things that affects what our sequencing is that it needs to be a clinical workflow. And so initially, Anna, who did a lot of this work, the workflow was to purify the RNA and DNA, amplify the RNA to cDNA, quantify and pool all the DNA together, and then do a library prep load and sequence for, say, 24 hours, and then do data analysis. But that workflow is a 3-day workflow. And remember, if this is eventually supposed to be a clinical test that could be used clinically, it's important to think about a workflow that actually could have meaning over existing diagnostics, right? Where a culture takes only 2 days to grow, for example. So one of the things that's necessary is to think about your time that you actually allow the DNA to be on the sequencer. And so this is data from one of the specimens with rhinovirus that detected rhinovirus strain 92. And within 30 minutes of sequencing, there were 10,000 reads, and the coverage rest was about 57%. Now, this is a virus that only has about 8,000 base pairs in its genome, so getting coverage rest of 57% may not be that hard in 30 minutes. We let the sequencer go for 48 hours, we get 2 million reads and then a coverage of closer to 100%. And so optimizing time, we ended up selecting that overnight might be kind of the right amount for RNA sequencing, and on a matter of, say, 6 to 12 hours for DNA sequencing. So here's how we improved the workflow with a shorter, someone just put a text. Do we typically only use the pneumonia biofire panel for BAL, or do we use it for other specimen types? Thank you, Hannah. The question is, we only use the pneumonia biofire panel for BAL specimens. There is also a biofire respiratory pathogen panel that is a nasopharyngeal swab. We do not typically do it from sputum, though I have known labs who have offline validated one or both of those panels for sputum. Is DNA from these rare pathogens not included in the biofire expected to be detectable levels in BAL during true infection? That is a good question, Brady. The answer is we hope that a true infection would have a predominant amount, or at least a large amount, or at least a good percentage of coverage of the pathogen genome, say 1% of that pathogen's genome for a bacteria. But as we're being able to detect things that we couldn't grow in culture now, it gets harder to define what the true pathogens are, actually. One of the problems, right, that you have with molecular assays is that they're more sensitive often than culture conditions. And so finding your reference data, it becomes more difficult. So the improved workflow for, someone else had one other. Very cool. Thanks. Oh, yeah. So one of the things to try to make this into a potential turnaround time that would work for a clinical assay was a pretty brilliant idea by Anna in the lab, who again decided that maybe we could use the same flow cell twice. Now, these flow cells range in price between, say, $600 and $1,000 a piece. And so maybe we could wash the flow cell and use it twice, which has been described. So but doing the library prep and loading of the DNA, because that's just purified as DNA, letting the DNA sequencing go, then washing the flow cell, all while the RNA to cDNA amplification is occurring, which takes about 6 hours, allowing then the RNA sequencing to occur overnight, and then the sort of sum analysis of both reactions in the AM. So that's how ones that were sequenced with both RNA and DNA were sequenced for what I showed before. So one of the other things that we had to optimize is the materials cost. And so currently this is what our materials cost. So the DNA purification and concentration reagents are $140 per test. So basically whether for RNA and DNA together. cDNA creation, which obviously is required for the RNA part, is $15. The flow cell and the library prep, we actually got a pretty good deal from Nanopore, and it's at $625. It was better than I thought. And then currently our computer analysis is free, though that may change because we're using currently Chan Zuckerberg Biohub software. And if you're going to do this as a clinical assay, we may not be able to use that software for our analysis. So we may have to do a software purchase or an internal software use. But currently the reagents are $780 per test. That does not include the tech time to spend basically an entire day doing DNA purification, DNA sequencing, RNA to cDNA preparation, flow cell lighting ×2, RNA sequencing overnight, and analysis the next day. So how does this assay so far potentially compare? So we're at this point around 36 hours from sort of the specimen receipt, which would be kind of when we spike or thaw out the specimen or receive it. And obviously our turnaround time is exactly the same as our receipt reported because we're not sending the specimen to anywhere. So far our materials are $780 and our labor is to be determined, but certainly that's in line at least with the cost of the University of Washington test. And certainly faster than the other tests that we're sending out, and maintains the ability to potentially detect DNA and RNA pathogens. So far, we've had a couple Aspergillus galactomannan positives that didn't grow anything in culture that weren't detected as Aspergillus. So I don't know whether our fungal pathogen detection will in the future be limited, but I put it here as a possible caveat. Okay, so a couple of interesting things. So one surprise happened was a surprise detection of DNA of HSV1 in one of our specimens. So when you're thinking about an accuracy analysis, you ask yourself, did I detect, was I expecting to detect? Later, when you think about precision and specificity, you're going to ask, did I detect surprise things? And in an ideal assay development, you don't necessarily want that, but obviously in molecular assays, especially for emerging pathogen detection, we kind of want some of that threshold. So we did have one surprise herpes simplex virus 1, H simplex human alpha 1 in a DNA from one of our specimens. And this patient was deceased when the specimen was transferred to the for this research. And so we were able to ask and find out that the patient had a known history of HSV, whether that contributed to their death, whether they were having an HSV outbreak in their mouth at the time. Don't know that. This isn't a clinical that kind of study currently, but it is interesting that we would not have found this HSV in this BAL with any of our standard methodology unless we were doing culture. And then for our rhinoviruses, for the one we got the most DNA sequence from, we had a contig of 1,300 bases. And then blasting that, it looked like it was rhinovirus B92. So there are hundreds of rhinoviruses out there. And typically people don't care about typing them, right? You just say it's a rhinovirus, it causes the common cold, you're going to get better. Nobody really dies of rhinoviruses. But similarly for, say, a coronavirus that was a novel coronavirus, or even influenza viruses, you potentially could have the information at your hands to maybe get at least typing of your virus done, maybe not the clonality you need, like we needed to look at COVID here at UCSD, that was done in other labs, but at least the ability to potentially get the typing, and you could say, wow, we're having an outbreak of rhinovirus 92, potentially B92 in this case. One last question that we've been asking since we've kind of struggled through optimizing the nanopore sequencing was whether or not we should have used Illumina sequencing. So there's one other lab that I know of doing sequencing, non-biased sequencing from BAL, and it's in China. And they compared Illumina sequencing to nanopore sequencing to culture and just said, what were the amount of specimens that we got counts? And they found that the nanopore sequencing was sort of more sensitive in their methodology, and in particular seemed to be more sensitive for viruses. Somebody says, this seems, well, I'll answer your question in just one second, Hannah. So that this and also for mycobacteria, and it was pretty amazing how few of the sort of culture positive bacterial pathogens they detected in their sequencing. And so they're kind of making strides in the same direction as we are, and maybe a little further along, and seem to argue that maybe we didn't choose wrong by using a longer read sequencer like the nanopore, not just for turnaround time reasons and cost per test reasons. Okay, so when NGS comes up positive for multiple pathogens, how do we make sense of what pathogen is driving the infection? Even pathogens that are very common commensals can cause pneumonia. So how do we determine the smoking gun so we can initiate treatment? Very good question. That requires really a clinical validation. And step one is really just to work on the analytic validation of saying, can we detect what's there? And how reliable is the detection? This is a question that people are asking. And like I said, the University of Washington, when they just did 16S sequence, found a bunch of anaerobes, and then went back and said, wow, in certain patients, there's a lot of data that suggests some of these anaerobes are making beta-lactamases that are inactivating antibiotics that might make it harder to treat the aerobes that are driving the infection, particularly in cystic fibrosis patients. So determining what beyond the known existing pathogens is important is difficult. And I think all we can say is there's a lot of it or it exists at this point. So next generation sequencing is able to detect some pathogens that we've spiked in. Our bacterial DNA pathogens were most robust. We were able to detect rhinovirus in a lot of specimens. The other RNA viruses, at least so far, and spiked in sequences have looked okay, but we haven't really done that validation yet. Bacterial DNA was detected that would not have been detected by current lab methods, such as lactobacillus species, again, an oral anaerobe that may not be doing much. Mycoplasma species, there are mycoplasma in the world. They're very hard to grow. There's certainly a few that cause disease, like Mycoplasma pneumonia, Mycoplasma hominis in the right patients, Mycoplasma genitalium in the right patients. And so, certainly detection of difficult to detect species we were able to show. We also detected a DNA virus that otherwise would not have been detected, HSV1. Again, whether it contributed to any pneumonia in this patient is hard to know, but it does make me wonder whether the patient also reactivated HSV as they were, in their clinical course. There are a large amount of human reads, and we're still struggling with this. We tried saponin and DNase, and it didn't really help reduce our human reads, so I'm not showing that data. We did have a faster computer that helped with some of the real-time exclusion. Tobacco mosaic virus contaminated a bunch of our specimens, which those results are not included today. Otherwise, I would show you a page of tobacco mosaic virus detection, but we were able to detect it multiple times. And then spiked inactivated viruses and spiked viral DNA were poorly compared to the positive just clinical bacterial specimens, probably due to the bacterial size. So the goal is to continue to do validation with positive clinical specimens, and we're expecting, of course, influenza and parainfluenza and all of the viruses to start peaking, starting imminently, actually, within a month or so. So our future direction is analytical sensitivity validation with DNA pathogen, RNA pathogen, and combined specimens with the focus on really the viruses, and then inclusion of mycobacteria, because that's a bacteria that can be really hard to diagnose because of its growth conditions and culture time. And then to ideally do a pilot experiment with ICU specimens specifically, where we know a little bit more about the specimens than just what was in them. What's needed even to bring this assay online is a lot more. So we're working currently on accuracy with clinical and simulated specimen samples. That's been really the focus and continues to be the focus of what the Prepare Award was by the flow cells, to optimize and see how accurate we can get it. But in order to bring an assay online to the lab, we need LOD studies. What UC San Francisco did was make a list of positive controls that I think are kind of becoming the reference list. And their list was CMV, HIV. They wanted a DNA and an RNA virus, Group B strep, Klebsiella pneumonia, Aspergillus niger for a mold, Cryptococcus neoformans for a yeast, and then Toxoplasma gondii for a parasite, a protozoa. For precision, you spend a number of cells on detection of over 20 consecutive positive control runs to look for intra-assay, and then inter-assay, you do the same run. Stability is important. You can't really bring a test online without knowing how stable your specimens are. And since this test is really not, this isn't just validating an FDA cleared test, the way that the other labs have done this is to take the positive control organisms, hold them for 0, 2, and 6 days, and then see if you can still detect them, and then also subject the specimens to freeze-thaw cycles. We've already seen reduction in sensitivity of the viruses, the RNA viruses that we were provided by providers, even just after a couple of freeze-thaw cycles. So it definitely is important. And then interference, the way that other labs have done it is to include the positive controls and then spike in a high concentration of either one of those organisms' DNA or an additional organism DNA or RNA or hemolytic blood, and make sure you can still detect the positive control organisms. This is important because we know that when we had a lot of tobacco mosaic virus, it definitely made our ability to detect some of the other viruses less. I'd like to acknowledge the people who've really been working on this. So David Pride has been my mentor in this and really has the lab space here at the Palms and in his lab on campus. Dr. Kobe Ngwemis, who's working in his lab, has really done the majority of the purification, the sequencing, the bulk of this work. Andrew Garcia, Alicia, and Monica, who is suddenly in black and not blue for reasons I don't know, are also members of the Pride Lab who've been instrumental in working on this project. And then Dr. Karlin Lewinsky and Nicole Steinmetz, or Dr. Steinmetz, for the RNA viruses they provided, and then the Prepare Institute for the funding for the flow cells and the reagents, which is where that came from. I'll leave it now for questions. Make myself so I can see.
Good job. I think there might have been some questions also that were in the chat that you might want to just.
Oh, did I miss
Did you
them?
get all those? I don't know. I'm just making sure.
I think I got them, but let me get to the chat here. I didn't see any more. Am I missing one?
Okay, that's good.
Yeah, I don't know if there are others.
I have a question,
Yeah.
Marcelo from JCVI. This pipeline is super interesting, especially that you can get back to the clinician so quick. I'm assuming quickly, and that's the question.
Yeah.
Can we talk about the time frame and how to optimize the time frame for this type of work? I believe this is key here for the future of this pipeline.
Yeah, so I will tell you what it causes issues with the currently available on the market things. So one of the biggest things is that they're not interfaced in our electronic medical record. So that means even when we receive things back, it has to be like manually faxed. So here you have this next generation sequencing technology that is like, blowing, diagnostics out of the water, and, they're not, and there's a lot of reasons for that. It's complicated, and the specimens have to be sent. And then I think also they are batching and bulking. And so I think Carius probably has the best reliable turnaround time from the time they receive the specimen. They've spent the most development on that. Here and in-house, you don't have that transfer issue, and we potentially have the ability to directly upload a result into our own EMR. So from a on-site development for UC San Diego patients, I think that the issues with turnaround time that sort of relate to the logistics, which is really one of the biggest things that happens, are going to be less, and it's really all about optimizing the assay. And so one of the key things to doing that is trying to have all of that sequencing running overnight, or at least running while you're preparing the cDNA library, which I think was a really brilliant idea by Anna in particular. So.
Okay, thank you.
Yeah.
Ahnika, I have questions and comments. Thank you so much. I'm so excited for this work as well, even though I'm not a clinician, but working in the same space for the fungal detection. We are more than happy to help with flow cell washes. I think Jake is also listening. So if you guys want to come over, we can show you, or we can come over to show flow cell washing as well. In addition, for the human sequence depletion, there are a couple kits, one of which we've used somewhat successfully. So there's Thermo Fisher's microbe enrich kit. This just removes the human, mouse, or rat sequences, which you can give it a shot. Obviously, that drives up the cost. The other one that I've seen is Deplete X. And additionally, if this becomes a pipeline, you can always order the Agilent's custom biotinylated probes to deplete the human sequences. That's a little bit upfront cost, but it then becomes cheap per sample. And what else? Oh, regarding your accuracy, I believe you said you use R9 flow cells, which should be Q20 chemistry, so it should be 99.7% accurate, because Nanopore has improved on those flow cells.
Yeah, I think what I meant was accuracy of detection of the actual pathogen that we knew was there, as opposed to accuracy of sequencing, because the flow cells are, yeah.
Yeah, I'm referring to the
yeah,
first
yeah.
slide that you showed in comparison. Like right now, both nanopore and PacBio are 99% greater than 99%.
Oh, yes, sorry, yeah, that initial slide with the comparator was, what's it called, was I stole it from an article that probably wasn't comparing the most current up-to-date nanopore flow cell, which had improved.
Yeah.
Yeah,
happy to help anytime. We can discuss obviously offline. And yeah, let us
the
know.
human reads have been a big issue because they just predominate no matter what, and so far we haven't really been able to reduce them a lot. So
yeah.
And what other, I think the reason other people haven't moved into the BAL space is because of just sort of the total amount of noise in the samples. And so, what like most labs have done is just said, okay, we're just going to target 16S so that we know we're working with just the bacteria. And so University of Washington has done that. But I think novel pathogens are likely to be RNA viruses. And so that's the part that sort of remains to do here, and the most, I think, most useful. Otherwise, might as well just sequence the bacterial reads, right?
Yeah.
So.
Okay, good stuff. Thank you, Ahnika. Thanks, everybody. What's our next seminar, David? I see you pulled up a slide. Oh, October 20, next one's in October. Carrie Byington's going to do it. She's now back at UCSD at her stint, after her stint being a big leader at UCOP, which is super happy that we have her back. So please don't miss it. It's going to look at how the UC system, COVID response, action report, and how we have lessons learned, etc. So should be pretty good. But we don't have the actual date. Is that right, David?
Yes, that's right. We don't have the date nailed down. It'll be probably in late October.
Okay, look for those announcements, and then everybody take care.